In October 2020, a small six-wheeled robot working alone on the floor of a Martian crater drilled a hole into a piece of ancient rock, collected a pinch of the resulting powder, and sealed it inside a small metal oven built into its own body. It would take nearly 6 years for anyone to fully understand what that powder actually contained. This is not a story about a sudden discovery. There was no dramatic moment of realization, no single instant when a scientist looked at a screen and gasped. It is instead a story about patience, about a machine built with such careful, deliberate foresight that it could capture a signal from 3.5 billion years in the past, store it faithfully, and wait for years until the right combination of instruments, techniques, and careful analysis finally allowed that signal to be properly read. What it eventually revealed, announced formally in April of this year, is the most diverse collection of organic molecules ever found on Mars. More than 20 individual compounds, 7 of them detected anywhere on the planet for the very first time. Among them, a ring-shaped molecule containing nitrogen, a structural cousin, in the loosest possible sense, of the molecules that form the backbone of DNA here on Earth. I want to be honest with you immediately about what this does and does not mean, because this particular story has a habit of being oversold, and this channel does not do that. This is not proof of life on Mars. It is not even strong evidence of life on Mars. What it is, carefully and precisely, is evidence that the chemistry required for life, the specific molecular building blocks that here on Earth eventually assembled themselves into something that could think and grow and one day build a rover and send it to another planet, was present on Mars, preserved in rock for the better part of 4 billion years. That is a genuinely extraordinary thing to have confirmed, and it sits alongside another discovery announced in the very same month that reshapes an entirely different question about the red planet. Not whether it could once have supported the chemistry of life, but whether it once had something almost unimaginable sitting on its surface. An ocean. Not a lake, not a river. An ocean, potentially covering a third of the entire planet, its ancient shoreline still faintly, patiently traced into the Martian landscape today in the shape of what researchers are calling, with a certain irresistible plainness, a bathtub ring. Tonight, we are going to sit with both of these stories, the patient chemistry and the vanished ocean, and we are going to ask honestly what they do and do not tell us about whether Mars once briefly in the deep past might have been alive. To understand what Curiosity actually found, you first need to understand the specific instrument responsible for finding it, and the very particular, very patient kind of chemistry it was built to perform. Curiosity is not simply a camera on wheels. Bolted inside its body is a genuinely remarkable piece of miniaturized laboratory equipment called SAM, the Sample Analysis at Mars Instrument Suite. SAM is, in essence, an entire analytical chemistry laboratory, the kind you might expect to find occupying a full room at a university, compressed down to fit inside a machine that had to survive a rocket launch, a 7-month journey through space, and a landing sequence so complicated that NASA engineers, only half-jokingly, nicknamed it 7 Minutes of Terror. SAM's job, since Curiosity landed in Gale Crater in August 2012, has been to search specifically for organic molecules, carbon-based compounds that, on Earth, form the essential chemical scaffolding of every living thing. Carbon is an unusually versatile atom, capable of bonding with itself and with other elements in long chains, branching structures and rings, producing molecules of extraordinary complexity. This versatility is precisely why life on Earth is built from carbon chemistry, and precisely why the search for organic molecules on Mars has been one of Curiosity's central scientific priorities from the very beginning of its mission. The challenge from the outset was not simply finding carbon-containing molecules, it was finding them intact. Mars's surface is bathed in a level of radiation far more intense than anything experienced on Earth's own well-shielded surface, since the planet lacks both a protective global magnetic field and a substantial atmosphere. Over billions of years, that radiation should in principle break down and destroy exactly the kind of delicate organic molecules Curiosity was sent to look for. Finding them intact and identifiable after 3.5 billion years of exposure was never going to be straightforward. For much of Curiosity's mission, the specific technique used to search for organics involved heating rock samples to extremely high temperatures inside SAM's small internal ovens, vaporizing whatever organic material might be present so that other instruments could analyze the resulting gases. This method worked, and it produced genuine results, including in 2018, the first confirmed detection of organic molecules preserved in Martian mudstone atoll, and later, in a study released in March 2025, the discovery of the largest organic molecules found on Mars to date, 3 long-chain hydrocarbons named decane, undecane, and dodecane, drilled from a rock called Cumberland in an area known as Yellowknife Bay, thought to be the fossilized bed of an ancient lake. But this heating-based method has a significant limitation, one that had frustrated the mission's own scientists for years. The intense heat required to vaporize the rock sample also tends to break larger, more complex organic molecules apart into smaller fragments, destroying much of the very structural information scientists most wanted to preserve and examine. It was, in a sense, like trying to identify a piece of intricate machinery by melting it down and analyzing the resulting puddle of metal. You could tell certain things, you could not tell everything. In October 2020, at a specific location in Gale Crater's Glen Torridon region, nicknamed Mary Anning after the pioneering 19th century English fossil hunter whose careful, patient work along the cliffs of southern England transformed the young science of paleontology, Curiosity drilled a rock sample and tried something different. This time, instead of simply heating the sample, Sam performed what is called a wet chemistry experiment, mixing the powdered rock with a chemical called TMAH, tetramethylammonium hydroxide, before heating it. This specific chemical process, known as thermochemolysis, is gentler and more selective than heating alone. It breaks down larger, more complex organic macromolecules in a controlled, predictable way, releasing smaller, identifiable fragments while preserving considerably more information about the original molecule's underlying structure than a simple heating experiment ever could. It was the very first time this specific technique had ever been attempted on the surface of another world. The rock sample was analyzed. The data was transmitted back to Earth, across the tens of millions of kilometers separating Mars from home. And then, for a considerable stretch of time, the specific results sat within that data, waiting for scientists to work through the painstaking process of properly, carefully interpreting what Sam's instruments had actually detected. The specific results of that October 2020 experiment were finally published in April 2026 in a paper in the journal Nature Communications, and they were worth the wait. The rock sample came from a specific geological layer called the Knockfarrel Hill member, part of the Glen Torridon region on the lower slopes of Mount Sharp, the towering central peak that rises from the middle of Gale Crater. This particular rock formed approximately 3.5 billion years ago, and its specific composition, fine-grained clay-rich sandstone, indicates it was deposited in a genuinely calm, stable, water-rich environment. Not a violent flood, not a brief, transient puddle, a persistent body of standing water, quiet enough and long-lived enough for fine sediment to settle gently to the bottom, layer upon layer, over an extended period of time. Clay minerals, specifically, are of particular interest to astrobiologists because they form through the sustained chemical interaction between rock and liquid water, and because they are known from extensive study here on Earth to be unusually effective at trapping and preserving organic molecules, protecting them from some of the destructive processes that would otherwise break them down over geological time. Finding a thick layer of ancient water-formed clay was, in itself, encouraging. Finding it to still contain a rich, readable chemical record within it was something considerably more significant. The wet chemistry experiment performed on the Mary Anning sample identified more than 20 distinct organic molecules. This is, by a clear and substantial margin, the most chemically diverse collection of organics ever detected in a single Martian sample. 7 of the specific molecules identified had never been detected anywhere on Mars before, in any previous sample, by any previous experiment. Among the compounds identified were nitrogen-bearing molecules, organic structures incorporating nitrogen atoms directly into their molecular framework, a category of chemistry considered particularly significant because nitrogen is a required ingredient in several classes of biologically essential molecules here on Earth, including amino acids, the building blocks of proteins, and the nucleobases that form the genetic alphabet of DNA and RNA. The collection also included aromatic compounds, ring-shaped molecular structures, and specifically a molecule called benzothiophene, a sulfur-containing aromatic compound. Perhaps the single most striking individual find was a specific nitrogen heterocycle, a ring-shaped molecular structure incorporating a nitrogen atom directly within the ring itself. This particular class of molecular architecture is structurally significant because it closely resembles the fundamental molecular scaffolding used by nucleobases, the specific chemical letters adenine, guanine, cytosine, and thymine that spell out the genetic code within every strand of DNA on Earth. To be entirely precise and careful about the claim being made here, this does not mean scientists found DNA, or even a direct precursor to DNA, on Mars. It means they found a molecule that shares a structural family resemblance with the specific class of chemistry from which DNA's own components are built, evidence that the kind of nitrogen-based ring chemistry life depends on was chemically achievable and was actually occurring in ancient Martian water. The research team's own interpretation, stated carefully in their published paper, is that this diverse suite of molecules most likely represents fragments of larger, more complex organic macromolecules that were originally present in the ancient lake sediment and have been slowly partially broken down over 3.5 billion years of burial, radiation exposure, and gradual geological transformation, the same slow process called diagenesis, by which loose sediment gradually becomes solid rock. What makes this discovery genuinely significant is not any single molecule in isolation, but the collective diversity of the whole assembly. A single organic molecule found in isolation might plausibly be explained by any number of mundane, non-biological chemical pathways, or could conceivably have arrived at Mars from an outside source entirely, carried in by a meteorite or comet impact. A rich, chemically diverse suite of more than 20 distinct organic molecules, including several classes of nitrogen chemistry considered biologically significant here on Earth, is a rather more striking and more difficult signal to explain away. As the Curiosity mission's own project scientist noted following the announcement, seeing this degree of chemical diversity suggests we may only be glimpsing a small fraction, the visible tip, so to speak, of a considerably greater diversity of organic chemistry that was actually present on ancient Mars. Here is where this particular story requires real scientific discipline, and where the temptation to overstate the finding must be resisted directly and honestly. Organic molecules, on their own, are not evidence of life. This is a genuinely important distinction, and one that is easy to lose sight of amid genuinely exciting news coverage. The word organic in a strict chemical sense simply means a molecule built around a carbon backbone. It does not mean the molecule was produced by a living organism. Organic molecules can, and routinely do, form through entirely non-biological chemical processes, geological reactions occurring deep within rock, chemical processes occurring in the cold vacuum of interstellar space, or reactions triggered by exactly the same kind of meteorite and asteroid impacts that regularly deliver material to planetary surfaces throughout the solar system. We touched on this directly in an earlier chapter of this series. Complex organic molecules, including amino acids, have been found preserved inside meteorites that fell to Earth, having formed somewhere out in space long before they ever encountered our planet, entirely independent of any biological process whatsoever. Mars, throughout its own long history, has been struck by countless such meteorites, each one a potential, entirely non-biological source of at least some fraction of the organic material Curiosity has detected. The research team behind the April 2026 findings was explicit and careful on precisely this point. The specific experiment performed by Curiosity's SAM instrument cannot, by itself, distinguish between organic molecules produced by ancient Martian biology and organic molecules produced through purely geological or chemical processes, entirely without the involvement of any living organism at all. The wet chemistry technique that finally allowed these molecules to be identified and characterized in such useful detail was never designed to make that specific distinction. It answers a different, though closely related question. Not was there life, but was the necessary chemistry there? There are broadly several plausible non-biological explanations for how this specific chemical diversity could have arisen on ancient Mars. Certain geological processes involving reactions between water, rock, and dissolved minerals under specific temperature and pressure conditions are known to be capable of synthesizing simple organic molecules directly, entirely without biology, a process sometimes referred to as abiotic organic synthesis. Some fraction of the detected material could plausibly have arrived via meteorite impact, as discussed a moment ago, and some fraction could represent the partial breakdown products of larger organic molecules that were themselves originally produced by any combination of these entirely non-biological pathways. None of this diminishes the genuine scientific significance of the discovery. It simply, honestly reframes precisely what has actually been established. What the April 2026 findings confirm with real confidence is that ancient Mars possessed the specific raw chemical ingredients and evidently the necessary environmental conditions: standing liquid water, clay-rich sediment, a chemically active environment, capable of producing and then preserving genuinely complex organic chemistry for an extended period in a stable enough setting for that chemistry to be recorded, essentially undisturbed, for 3.5 billion years. Whether that chemistry ever crossed the line into anything we would recognize as biology remains, honestly and entirely, an open question. It is a question this specific experiment was never designed to resolve, and it is a question the scientists themselves are careful never to claim they have resolved. While Curiosity's own team was completing the careful analysis that would eventually produce the April 2026 organics paper, an entirely separate group of researchers was working on a different, though closely related, Martian mystery, one operating at a vastly larger physical scale. For decades, planetary scientists have debated a genuinely fundamental question about Mars's ancient past. Did the planet, roughly 3 to 4 billion years ago, when its atmosphere was thicker and its climate considerably warmer than the frozen desert we observe today, possess not merely scattered lakes and rivers, but an actual ocean, a large standing body of water, covering a meaningful fraction of the planet's entire surface, comparable in basic concept, if not in exact scale, to Earth's own oceans? The idea is not new. It dates back to the 1970s, when NASA's twin Viking orbiters first returned images suggesting the presence of an ancient shoreline traced across Mars's northern lowlands, together with a large ocean-sized depression in the planet's overall northern topography. But the specific evidence has remained persistently ambiguous and difficult to fully confirm. Proposed shoreline features, examined closely over subsequent decades, have often turned out to sit at inconsistent varying elevations across different regions of the planet, a serious problem for the ocean hypothesis, since a genuine, stable body of standing water should, by basic physical necessity, leave a shoreline at a single geologically consistent elevation, exactly as Earth's own oceans do. In April 2026, a research team led by Abdollah Zaki, a postdoctoral researcher at the University of Texas at Austin, together with Caltech geology professor Michael Lamb, proposed an entirely new way of approaching this long-standing question. Rather than searching directly for a fragile, ambiguous shoreline, a feature that would, after billions of years of wind erosion, dust deposition, and general geological weathering, likely be extremely difficult to positively identify with any real confidence, the team instead searched for something considerably more durable and more geologically robust: a continental shelf. On Earth, a continental shelf is the relatively flat, gently sloping band of underwater terrain that surrounds a continent's coastline before the sea floor eventually drops away more steeply into open, deep ocean. Even as sea levels rise and fall over long stretches of geological time, causing the exact position of any specific shoreline to shift considerably, a continental shelf itself tends to remain a comparatively stable, persistent, large-scale landform, tracing the fundamental transition zone between land and ocean across enormous spans of geological time. Using detailed topographic data of the Martian surface, originally collected by an orbiting laser altimeter instrument, Zaki and Lamb searched specifically for an analogous large-scale feature somewhere within Mars's northern lowlands. They found one, a broad, distinctly flattened band of terrain, in places several hundred kilometers wide, tracing a long, continuous arc across the northern hemisphere of the planet, precisely the kind of large-scale geological signature a genuine, long-lived ocean's own continental shelf should be expected to leave behind once the ocean itself had long since evaporated or otherwise disappeared. Because this specific feature so closely resembles the ring of built-up residue left behind around a drained bathtub, tracing, in this specific case, the outline of where a genuine ancient sea once met ancient Martian land, researchers have taken, somewhat irresistibly, to informally calling it precisely that: Mars's bathtub ring. Supporting evidence for the interpretation arrived from an entirely independent line of research, examining ancient river delta deposits at two specific Martian locations, Aeolis Dorsa and Hypanis Vallis. River deltas form specifically where a river's flow meets a standing body of water, depositing sediment as its current slows and spreads out. Careful analysis of these particular deltas revealed clear evidence of significant sustained changes in local water level over time. In one location, a rise of some 900 meters, and at the other, a subsequent drop of roughly 500 meters, patterns consistent with meaningful sea level change occurring within a genuinely large standing ocean, rather than the kind of comparatively minor short-lived fluctuation you would expect from an isolated lake or seasonal pond. Taken together, the proposed continental shelf and the independently observed patterns of ancient delta sea level change form a mutually reinforcing picture. An ocean that at its largest extent may have covered as much as 1/3 of the entire surface of Mars. Remarkably, the same basic bathtub ring phenomenon, the durable, long-lasting geological trace left behind by a body of water long after the water itself has vanished, appears not only at the scale of an entire hypothesized Martian ocean, but at the vastly smaller, more local scale of Gale Crater itself, the specific location Curiosity has been slowly, patiently exploring since 2012. Within a specific rock layer partway up the slopes of Mount Sharp, called the Amapari marker band, Curiosity detected unusually high concentrations of three distinct metals, iron, manganese, and zinc, the highest levels of these particular elements recorded anywhere during the entire mission to that point. This specific enrichment immediately caught the attention of the mission science team because it represented a genuine geological anomaly. Metals of this kind do not typically become locally concentrated at elevated levels by accident. Under the right chemical conditions, they are known to precipitate directly out of standing liquid water, gradually settling and accumulating within a specific, narrow band of sediment as a lake's water level itself very gradually falls over an extended period, concentrating whatever dissolved minerals remain within it into an ever-shrinking, ever more mineral-rich volume of remaining water. Patrick Gazda, a member of the science team responsible for Curiosity's ChemCam instrument, led a detailed study of this specific metal-rich band, published as researchers continued working to understand precisely how and why these three particular metals had become so concentrated in this one specific narrow layer. The team's favored explanation closely mirrors the exact process by which a bathtub, as its water level slowly drops, leaves a visible ring of built-up residue and mineral deposit at each successive waterline. As the paleolake that once filled Gale Crater gradually dried out, evaporating slowly over an extended period, dissolved metals within the shrinking body of water became increasingly concentrated, ultimately precipitating out and settling into the specific sediment layer that would 3.5 billion years later become the Amapari marker band. This more local crater-scale bathtub ring offers something the larger continent-spanning hypothesized ocean currently cannot. An object Curiosity has directly physically driven across and sampled itself, rather than a large-scale feature currently inferred only from orbital measurement and remote topographic data. It provides direct on-the-ground confirmation that Gale Crater itself functioned for some considerable stretch of ancient Martian history as a genuine, long-lived, gradually evaporating lake, precisely the kind of stable, long-lived, chemically active watery environment in which the diverse organic chemistry described a few chapters ago could plausibly have formed and then crucially been preserved. Considered together, these two bathtub rings, one traced across an entire hemisphere from orbit, the other identified directly within the walls of a single crater by a rover's own onboard instruments, reinforce a broader, increasingly well-supported picture of ancient Mars, a planet that was not merely occasionally briefly damp, but one that, for some genuinely extended period of its early history, hosted standing bodies of liquid water at multiple different scales simultaneously, small, localized, evaporating crater lakes, and quite possibly a vast ocean-scale body of water spanning an entire third of the planet's surface. If we allow ourselves carefully and with appropriate scientific caution to combine these separate strands of evidence, the diverse organic chemistry discovered at Mary Anning, the crater-scale bathtub ring at the Amapari marker band, and the considerably larger hypothesized ocean-scale bathtub ring traced across the northern lowlands, what kind of picture of ancient Mars begins to emerge? The most straightforward, most conservative reading is this. Somewhere between 3 and 4 billion years ago, Mars possessed a considerably thicker atmosphere and a considerably warmer climate than it does today, sufficient to sustain liquid water at its surface for extended periods, potentially including an ocean covering roughly a third of the entire planet. Within at least some portion of that water, including quite specifically the ancient lake that once filled Gale Crater, chemical conditions were suitable for the formation and gradual accumulation of a genuinely diverse suite of organic molecules, including nitrogen-based ring compounds structurally related to the fundamental chemistry underlying life on Earth. This picture, on its own, describes a planet that was, for some meaningful window of time, chemically habitable, possessing the necessary liquid water, the necessary organic chemistry, and, from other independent lines of Curiosity's research conducted over the years, evidence of other chemical ingredients considered essential for life, including various usable energy sources and the specific chemical elements beyond carbon and nitrogen that biology on Earth depends upon. Whether ancient Mars ever actually crossed the specific threshold from mere chemical habitability into genuine, functioning biology is a categorically different and currently far more difficult question, one that current instruments, including Curiosity's own onboard SAM laboratory, are simply not capable of definitively answering, regardless of how much additional analysis is eventually performed on the specific rock samples already collected. This is precisely why NASA and its partner space agencies have spent years developing more ambitious follow-up efforts, specifically designed to push this particular question further. The Perseverance rover, currently exploring an entirely separate site called Gale Crater, has been specifically deliberately collecting and sealing rock samples in a series of small titanium tubes left behind at designated locations on the Martian surface, in careful anticipation of a future Mars sample return mission, an ambitious multi-spacecraft undertaking specifically intended to eventually retrieve those sealed samples and physically transport them back to Earth, where they could be examined using laboratory equipment far more sophisticated, far more sensitive, and considerably less constrained by mass, power, and complexity limitations than anything that could ever be sent to Mars aboard a rover. Separately, the European Space Agency's own Rosalind Franklin rover, expected to land on Mars before the end of this decade, will carry a drill specifically capable of reaching significantly greater depths below the Martian surface than Curiosity's own drill can access. An important capability, since material buried more deeply below ground would have been considerably better shielded from billions of years of surface radiation exposure, potentially preserving organic chemistry, if any biological signal genuinely exists to be found, in a rather more pristine, less degraded condition than anything accessible from Mars's heavily irradiated surface layer. And in a detail that connects this particular story to an entirely different world, discussed nowhere else in this series, NASA's upcoming Dragonfly mission, a rotorcraft specifically designed to fly across the surface of Saturn's moon Titan, will carry its own version of a wet chemistry experiment closely related to the very technique that finally allowed Curiosity's Mary Anning sample to reveal its full chemical diversity after 6 long years. Titan possesses its own genuinely exotic, low-temperature organic chemistry, built around liquid methane rather than liquid water, and scientists are eager to apply the exact same fundamental analytical approach that succeeded so well on Mars to an entirely different, considerably stranger planetary environment. None of these various efforts guarantees a definitive final answer regarding whether life ever existed on ancient Mars, but together they represent a coherent, patient, carefully sequenced scientific strategy methodically working to close the specific gap between the necessary chemistry was present and life was actually definitively there, a gap that, as of this particular month, remains genuinely, honestly open. There is something genuinely worth pausing on in the specific detail that the rock sample responsible for this entire month's headline discovery was collected at a location Curiosity's own science team had chosen to name Mary Anning. Mary Anning was born in 1799 in the English coastal town of Lyme Regis into a family of quite modest means. From a remarkably young age, she began searching the eroding coastal cliffs near her home for fossils, initially to sell to visiting collectors as a way of helping to support her family financially, in an era considerably before paleontology had become established as any kind of formal, professionally recognized scientific discipline. Anning's specific talent for locating, carefully extracting, and correctly interpreting fossils was genuinely extraordinary. She is credited with discovering the very first correctly identified ichthyosaur skeleton as well as some of the earliest known plesiosaur fossils, ancient marine reptiles that had, until her careful, patient excavation work, been entirely unknown to formal science. Her discoveries provided some of the earliest, most compelling physical evidence for the specific idea that entire species had once existed on Earth and had subsequently gone fully extinct, a concept that was, at the specific time she was working, still genuinely controversial and far from broadly formally accepted within the wider scientific community. Despite the genuine, undeniable importance and rigor of her work, Anning herself was, throughout her own lifetime, largely excluded from the formal scientific institutions of her era, owing to a combination of her working-class background and, quite simply, her gender. She was not permitted to become a member of the Geological Society of London, despite the fact that many of the specific specimens she personally discovered and helped correctly interpret went on to form the essential foundational evidence for entirely new areas of the emerging science of geology and paleontology. Male scientists and collectors frequently purchased her specimens and subsequently published academic papers describing and interpreting them, often without providing her any meaningful formal credit or acknowledgement whatsoever. It is only in recent decades that Anning's genuinely foundational, essential contribution to the modern science of paleontology has been more widely, properly recognized and celebrated. There is a particular quiet resonance in the specific fact that a robot from Earth, patiently searching an alien planet for the preserved chemical and physical traces of ancient life, chose to name one of its own most significant discovery sites after a woman who spent her own entire life doing precisely, exactly the same fundamental kind of work here on Earth, carefully, patiently reading the deep story written into ancient rock, often working for years or even decades before the full, true significance of any single specific discovery was ever fully, properly recognized and understood. Curiosity itself embodies in its own specific way a very similar kind of patience. The particular rock sample at the center of tonight's discovery was collected in October 2020. The full, complete analysis of what that sample actually contained was not published until April 2026, nearly 6 full years later. In the intervening time, the rover continued its own slow, patient work, driving further across the Martian surface, drilling additional samples, gradually building an ever more complete picture of Gale Crater's own ancient deep past, while back on Earth, an entire dedicated team of scientists worked carefully and methodically through the difficult, genuinely painstaking process of properly analyzing, interpreting, and finally publishing what that one specific sample had to reveal. Science, at its very best, frequently operates on precisely this kind of timescale. Not the instant, dramatic breakthrough of popular imagination, but the slow, careful, cumulative work of patient people and patient machines, working steadily toward an understanding that may take years, or in Mary Anning's own case considerably longer still, to be fully recognized for what it genuinely represents. There is a particular way of thinking about Mars that I find myself returning to, again and again, when I sit quietly with everything this month's discoveries have actually revealed. For the greater part of human history, Mars has served in our collective imagination as a kind of blank, distant canvas, a small reddish point of light in the night sky onto which we have projected an enormous variety of stories, an ancient warrior god, a home to invading alien civilizations, in more recent decades a potential future refuge for humanity itself, should our own planet ever become genuinely uninhabitable. Almost all of those particular stories were built from imagination alone, in a near-total absence of any real direct evidence about what Mars actually was or ever had been. What curiosity and perseverance and the various orbiting spacecraft studying Mars from above have gradually patiently assembled instead, particularly over just the past 2 decades, is something rather more remarkable than any of those older imagined stories, and rather more strange, direct, physical, scientifically grounded evidence that Mars, once, billions of years ago, may genuinely have resembled a considerably younger, considerably wetter version of Earth itself. A planet with rivers, with lakes, quite possibly if the specific bathtub ring evidence discussed earlier tonight continues to hold up under further scrutiny, with an actual ocean, covering perhaps a full third of its entire surface. A planet where liquid water sat in prolonged direct chemical contact with rock and with clay for long enough, and under the right conditions to produce a genuinely diverse suite of complex organic chemistry, the same fundamental category of chemistry that on our own planet eventually assembled itself gradually into every living thing that has ever existed, including, eventually, you. We do not know, and cannot currently know, whether that ancient Martian chemistry ever crossed the specific threshold into anything we would properly recognize as life. It may have, it may not have. The rock at Mary Anning cannot tell us on its own which of those two possibilities is true, and neither can the bathtub ring traced faintly across Mars's ancient northern lowlands. What both discoveries can tell us with real, grounded scientific confidence is that the fundamental question of whether ancient Mars was alive is not merely an idle speculative one. It is a serious, live, genuinely open scientific question, actively, carefully being investigated using real physical evidence, patiently gathered directly from the Martian surface itself. Consider for a moment what it would specifically mean if a Mars sample return mission, or the Rosalind Franklin rover's own considerably deeper drill, eventually turned up genuine, confirmed, unambiguous evidence of ancient Martian life, even the simplest possible microbial variety, long since extinct. It would mean life is not a singular, isolated, one-off event that happened exactly once, in exactly one place, in the entire observable universe. It would mean that, given roughly similar starting chemical and environmental conditions, life reliably tends to emerge, that biology, under the right circumstances, is less an extraordinary, singular cosmic accident, and rather more a fundamental, dependable tendency of chemistry itself, occurring wherever the right specific conditions happen to be met. And if instead, decades of increasingly careful, increasingly sensitive future investigation eventually determined with real confidence that ancient Mars possessed every single one of the specific chemical and environmental ingredients apparently required for life, and yet somehow never actually crossed that particular threshold, that would be, in its own quiet way, an equally profound and significant result. It would suggest that the specific chemical step from complex organic chemistry to genuine, self-sustaining biology is considerably harder, considerably rarer, and considerably more delicate than we might otherwise be inclined to assume. Either answer would meaningfully reshape a question we first explored together, all the way back in the opening episode of this entire series. Are we alone in the universe? Mars, sitting so remarkably close to Earth in purely cosmic terms, a short, comparatively modest hop across our own shared solar system, rather than any unreachable distance measured in light years, offers something no distant exoplanet currently can. A genuine, physically reachable place where we can actually go and drill and directly, carefully look for the answer ourselves, rather than merely inferring it indirectly from light gathered by a telescope many light-years removed from its ultimate source. Tonight, on the floor of Gale Crater, curiosity continues its own slow, patient work, exactly as it has for more than a decade now. It is not currently expecting any dramatic, singular, final answer to arrive. It is doing precisely what Mary Anning herself once did, patiently working the cliffs of an English coastal town nearly 2 centuries ago, reading rock, one careful, patient layer at a time, and quietly, steadily building, sample by sample, a story that may ultimately take considerably longer than any single human lifetime to finally fully understand. Somewhere out there, sealed inside a small metal drill tube, or waiting patiently within a rock formation curiosity has not yet even reached, there may be an answer to one of the single oldest questions our species has ever asked. We do not yet know precisely when it will finally properly speak, but we are still listening. And this month at least, Mars had considerably more to say than we had previously realized.